Hi class, this is your instructor, Scholar Huff. We're now in Chapter 30, Seed Plants. The history of land plants is characterized by evolutionary innovations that allowed the ancestors of aquatic algae to colonize and proliferate in harsh and varied terrestrial environments. Early innovations made survival during this transition to land possible. The subsequent explosion of plant life continues to change the land and atmosphere and supports terrestrial animal life. Over the last several hundred million years, seed-producing plants have come to dominate the terrestrial landscape. Much of this remarkable success of seed plants is due to the evolution of the seed, like the beans you see pictured here. Seeds protect the delicate embryo and provide food for them. Seeds allow embryos to pause development and growth and germinate after a harsh winter or dry season has passed. The latter innovation of fruits enhanced the dispersal of seeds across a wider landscape. Within the seed plants, the flowering plants have co-evolved intricate relationships with animals to enhance sexual reproduction as well as aiding the dispersal of the next generation. Numerous evolutionary solutions to terrestrial challenges have resulted over 400,000 species of seed plants dominating the all-terrestrial communities today. Collectively, these seed plants affect almost every aspect of our lives, from improving environmental quality to providing pharmaceuticals, food, fuels, building materials, and clothes. Seed plants appear to have evolved from spore-bearing plants known as progymnosperms. Progymnosperms shared several features with modern gymnosperms, including secondary vascular tissues, increasing in girth. Some progymnosperms had leaves. Their reproduction was very simple, and it was not certain which group of progymnosperms gave rise to seed plants. Biologists have long been intrigued by the rapid success and diversity of seed plants, particularly the diversification of angiosperms, or flowering plants. This diversity was made possible by the evolution of the seed, but it likely involved a number of innovations in different lineages. From an evolutionary and ecological perspective, the seed represents an important advance. The embryo is protected by an extra layer or two of sporophyte tissue called the integument, creating the ovule. Within the ovule, meiotic division occurs in the megasporangium, producing a haploid megaspore. The megaspore divides by mitosis to produce a female gametophyte, carrying the female gamete, an egg. The egg combines with the male gamete, a sperm, resulting in a zygote. The single-celled zygote divides by mitotic cell division to produce the young sporophyte, an embryo. Seeds also contain a food supply for the developing embryo. During development, the integuments harden to produce the seed coat, which protects the embryo. The seed is easily dispersed. Perhaps even more significantly, the presence of seeds introduces into the life cycle a dormant phase that allows the embryo to survive until environmental conditions are favorable for further growth. Seed plants produce two kinds of gametophytes, male and female, each of which consists of just a few cells. A consistent feature in the evolution of plants is a reduced size of the gametophyte and corresponding increase in dominance of the sporophyte generation. A pollen grain is a multicellular male gametophyte carrying the male gamete, a sperm cell. Pollen grains are carried to the female gametophyte by wind or by a pollinator. In some seed plants, the sperm moves toward the female gametophyte through a growing pollen tube. This eliminates the need for external water. In contrast to the seedless plants, the whole male gametophyte, rather than just the sperm, moves to the female gametophyte. A female gametophyte forms within the protection of the integuments, collectively forming the ovule. In angiosperms, the ovules are completely enclosed within additional diploid sporophyte tissue, and the ovule and the surrounding protective tissue are called the ovary. The ovary develops into the fruit. This is all the places we are about to go. Seeds distinguish the gymnosperms from the pterophytes. Gymnosperms encompass 4 of 5 lineages of seed plants. The 4 groups of gymnosperms are the coniferophytes, the cycadophytes, the gnetophytes, and the ginkgophytes. The 5th lineage of seed plants is the angiosperms. Although both gymnosperms and angiosperms produce seeds, only the angiosperms produce flowers and fruits. In gymnosperms, the ovule, which becomes a seed, rests exposed on a scale and is not completely enclosed by sporophyte tissues at the time of pollination. The name gymnosperm means naked seed because the seed develops on the surface of the scale. Gymnosperms vary in their reproduction and growth form. For example, cycads and ginkgo have motile sperm, whereas conifers and gnetophytes have sperm with no flagella. All sperm are carried within a pollen tube that forms when a pollen grain germinates. Most gymnosperms are terrestrial, although a few aquatic species exist. The most familiar gymnosperms are conifers, which include pines, spruces, firs, cedars, hemlocks, yews, larches, cypresses, and others. Nearly 40% of the world's forests are composed of conifers. The coastal redwood, a conifer native to northwestern California and southwestern Oregon, is the tallest living vascular plant. It may reach a height of nearly 100 meters, which is 300 feet. Another conifer, the bristlecone pine, of the White Mountains of California, the oldest living tree. One specimen is 4,900 years old. Conifers are found in cooler temperature and sometimes dry regions of the world. Various species are sources of timber, biofuel, feedstock, paper, resin, paclitaxel. Taxol is used to treat certain cancer, and other economically important products. Genomes of 3 conifers, the loblolly pine, the sugar pine, and Douglas fir, have been sequenced because of their economic importance. This was a substantial challenge because their genomes are about 10 times larger than the human genome. More than 100 species of pines exist today, all native to the northern hemisphere. Pines and spruces, which belong to the same family, are members of the vast coniferous forests that lie between the Arctic tundra and the temperate deciduous forests and prairies to their south. During the past century, pines have been extensively planted in the southern hemisphere. Pines have tough, needle-like leaves produced mostly in clusters of 2 to 5. Among the conifers, only pines have clustered leaves. The leaves, which have a thick cuticle and recessed stomata, or sunken stomata, represent an evolutionary adaptation for minimizing water loss. This strategy is important because many of the trees grow in areas where topsoil is frozen for part of the year, making it difficult for the roots to obtain water. The leaves and other parts of the sporophyte have canals which surrounding cells secrete resin. The resin deters insect and fungal attacks. The resin of certain pines is harvested commercially for its volatile liquid portion called turpentine, for the solid rosin, which is used on the bow hair of bowed stringed instruments. The wood of pine lacks some of the more rigid cell types found in other trees and is considered a soft rather than hard wood. The thick bark of pines is an adaptation for surviving fires, and sub-zero temperatures. Some cones actually depend on fire to open, releasing seeds to reforest burned areas. All seed plants produce two types of spores that give rise to two types of gametophytes, referring to being heterosporous. The male gametophytes, the pollen grains of pines, develop from microspores, which are produced by meiosis in male cones that develop in clusters, typically at the tips of lower branches. There may be hundreds of such clusters on any single tree. Male cones are typically clustered on the lower branches of pine trees, and female cones are often in the upper branches. Pines can produce so much pollen that it can sometimes be seen as a green mist above a fine forest. As the pollen rises from the lower branches of the tree on gentle breezes, it can pollinate female cones in the upper branches. The male pine cones generally are 1 to 4 centimeters long and consist of small papery scales arranged in spiral or whorls. A pair of microsporangia form sacs within each scale. Numerous microspore mother cells in the microsporangia undergo meiosis. Each becoming 4 microspores. The microspores divide by mitotic cell division to produce 4-celled pollen grains, each with a pair of air sacs that give them added buoyancy when released into the air. A single cluster of male pine cones may produce more than 1 million pollen grains. 2 ovules develop toward the base of each scale. Each ovule contains a megasporangium called a nucellus. The nucellus itself is completely surrounded by a thick layer of cells called the integument that has a small opening, the micropyle, toward one end. One of the cell layers of integument later becomes a seed coat. A single megaspore mother cell within each megasporangium undergoes meiosis, becoming a row of 4 megaspores. 3 of the megaspores break down, but the remaining one, over the course of about a year, slowly develops into a female gametophyte through mitotic divisions. The female gametophyte at maturity may consist of thousands of cells, with 2 to 6 archegonia formed at the micropylar end. Each archegonia contains an egg so large it can be seen without a microscope. Female cones usually take 2 or more seasons to mature. At first, they may appear reddish or purple in color, but they soon turn green, and during the first spring, the scales spread apart. While the scales are open, pollen grains are carried by wind drift between them, some catching in sticky fluid oozing out of the micropyle. The pollen grains within sticky fluid are slowly drawn down through the micropyle to the top of the nucellus, and the scales close shortly thereafter. The archegonia and the remainder of the female gametophyte are not mature until about a year later, when the female gamete is developing. A pollen tube emerges from a pollen grain at the bottom of the micropyle and slowly digests its way through the nucellus and the archegonia. During the growth of the pollen tube, one of the pollen grain's 4 cells, the generative cell, divides by mitotic cell division, with one of the resulting 2 cells dividing more than once more. The last 2 cells function as sperm. The germinated pollen grain, with its 2 sperm, is the mature male gametophyte, a small, simple haploid structure compared with fern gametophytes. About 15 months after pollination, the pollen tube enters the ovule through the micropyle and discharges its contents. One sperm unit unites with the egg, forming a zygote. This is the beginning of the sporophyte generation. The other sperm and cells of the pollen grain degenerate. The zygote develops into an embryo surrounded by a female gametophyte cells, which provide nutrition. The integument hardens to produce the seed coat. After dispersal and germination of the seed, the young sporophyte of the next generation develops into a tree. Cycads from Cycadophyta are slow-growing gymnosperms of tropical and subtropical regions. The sporophytes of most of the 250 known species resemble palm trees, with trunks that contain heights of 15 meters or more. Unlike palm trees, which are flowering plants, cycads produce cones and have a life cycle similar to that of the pines. Individuals of the species make either pollen-bearing or ovule-bearing cones, but not both. As such, members of the cycads could be considered to have separate sexes, being dioecious. The female cones, which develop upright among the leaf bases, are huge in some species and can weigh up to 45 kilograms. The sperm of cycads, although formed within a pollen tube, are released within the ovule and swim to the archegonium. Several species of cycads are facing extinction in the wild, and soon may only exist in botanical gardens. Chnidophytes from phylum Chnidophyta. There are 3 genera with about 65 extant species of chnidophytes. They are the only gymnosperms with vessels in their xylem. Vessels are a particularly efficient conducting cell type that is a common feature of the angiosperms. The members of the genera are greatly different from one another in form. One of the most bizarre of all plants is Welwitschia, which occurs in the Namib and Mozambique deserts of southwestern Africa, shown to you here. The stem is shaped like a large, shallow cup that tapers into a taproot below the surface. It has 2 strap-shaped leathery leaves that grow continuously from their base, splitting as they flap in the wind. The reproductive structures of Welwitschia are cone-like, appear toward the bases of the leaves around the stems, and are produced on separate male and female plants. And again, that means that it is dioecious. And this plant has some of the longest leaves on Earth. More than half of the chnidophyte species are in the genus Ephedra, which is common in arid regions of the western United States and Mexico. Species are found on every continent except Australia. The plants are shrubby, with stems that are superficially resemble the horsetails, being jointed and having tiny scale-like leaves at each node. Male and female reproductive structures may have been produced on the same or different plants. The drug ephedrine, widely used in the treatment of respiratory problems such as congestion, was in the past extracted from Chinese species of ephedra, but now has been largely replaced with synthetic preparations, pseudoephedrine, because ephedrine found in herbal remedies for weight loss was linked to strokes and heart attacks. It was withdrawn from the market in April of 2004. Sales restrictions were placed on pseudoephedrine containing products in 2006 because it can be used to manufacture the illegal drug methamphetamine. The best-known species of the third genus, Netum, is a tropical tree, but most species are vine-like. All species have broad leaves similar to those of angiosperms. One atom species is cultivated in Java for its tender shoots, which are cooked as a vegetable. And lastly, the ginkgophytes from Ginkgophyta. The fossil record indicates that members of the ginkgophytes from phylum Ginkgophyta were once widely distributed, particularly in the northern hemisphere. Today, only one living species, Ginkgo biloba, remains, shown to you all here. This tree, which sheds its leaves in the fall, was first encountered by the Europeans in cultivation in Japan and China. It apparently no longer exists in the wild. Like the sperm of cycads, those of ginkgo have flagella. The ginkgo is dioecious, that is, the male and female reproductive structures are produced on separate trees. The fleshy outer covering of seeds of the female ginkgo plants exude a foul smell of rancid butter. I say it's worse than that. I've smelled it more than once. Caused by the presence of butyric acid and isobutyric acids, which are also found in butter and vomit. I say it more smells like vomit. Because of its beauty and resistance to air pollution, ginkgo is commonly planted along city streets. However, due to the rancid smell produced by the female ginkgo plants, male plants produced by vegetative propagation are preferred for this use. Angiosperms are a group of seed plants comprising some 300,000 species of flowering plants. Unlike gymnosperms, the angiosperms have ovules that are enclosed within diploid tissue at the time of pollination. The carpel, a modified leaf that encapsulates seeds, develops into the fruit, a unique angiosperm feature. Although some gymnosperms, including the yew, have flesh-like tissue around their seeds, it is of different origin and not a true fruit. Today, there are some 300,000 species of flowering plants. Only about 1,000 species of all other seed plants combined. So the unique features of these, called angiosperm, aided in their abundance. Flower production is one of those. The ability to attract insect pollinators, secondly, and the development of broad leaves with dense veins. So this shows the seed plant cladogram, beginning back with Ginkgophyta, the netophytes, conifers, and cycads, and then getting to the Archaefructus, which is extinct, the Amborella, water lilies, the star anise, eudicots, and the magnoliids, and then the monocots lastly. Flowers are considered to be modified stems bearing modified leaves. Regardless of their size and shape, they all share common features. Each flower originates as a primordium that develops into a bud at the end of a stalk called a pedicel. The pedicel expands slightly at the tip to form the receptacle to which the remaining flower parts are attached. The other flower parts typically are attached in circles called whorls. The outermost whorl is composed of sepals. Most flowers have 3 to 5 sepals, which are green and somewhat leaf-like. The next whorl consists of petals, often colored, attracting pollinators such as insects, birds, and some small mammals. The petals, which also commonly number 3 to 5, may be separate, fused together, or missing altogether in wind-pollinated flowers. At the center of the flower is The third whorl consists of stamens and is collectively called the androecium. This whorl is where the male gametes, pollen, are produced. Each stamen consists of a pollen-bearing anther and a stalk called a filament, which may be missing in some flowers. At the center of the flower now is the fourth whorl, the gynoecium, or some may say the gynoecium, where the small female gametes are housed. The gynoecium consists of one or more carpels. The first carpel was derived from the leaf-like structure with ovules along its margins, and then primitive flowers can have several to many separate carpels, but in most flowers, 2 to several carpels are fused together. Such fusion can be seen in an orange sliced in half. Each segment represents one carpel. A carpel has 3 major regions. The ovary is the swollen base, which contains from 1 to hundreds of ovules. The ovary later develops into a fruit. The tip of the carpel is called a stigma. Most stigmas are sticky or feathery, trapping pollen grains that land on them. Typically, the neck or stalk, called a style, connects the stigma to the ovary. In some flowers, the style may be very short or even missing. Many flowers have nectar-secreting glands, called nectaries, often located toward the base of the ovary. Nectar is a fluid containing sugars, amino acids, and other molecules that attracts insects, birds, and other animals to flowers. During development of a flower bud, a single diploid megaspore mother cell in the ovule undergoes meiotic cell division, producing 4 haploid megaspores. In most flowering plants, 3 of the megaspores soon disappear. The nucleus of the remaining megaspore divides mitotically, and the cell slowly expands until it becomes many times its original size. While the expansion of the megaspore is occurring, each of the daughter nuclei divides twice, resulting in 8 haploid nuclei arranged in 2 groups of 4. One nucleus from each group migrates toward the center, where they function as polar nuclei. A cell wall forms around the 2 polar nuclei to create a central cell. Cell walls form around the remaining nuclei. In the group closest to the micropyle, one cell functions as the egg. The other 2 cells are called synergids. At the other end, the 3 cells are now called antipodals. They have no apparent function and eventually break down and disappear. At the same time, 2 layers of the ovule, the integuments, differentiate into what become the seed coat of a seed, and the integuments, as they develop, form the micropyle, a small gap or pore at one end that was described a bit earlier on. The large sac with 8 nuclei and 7 cells is called an embryo sac. It constitutes the female gametophyte. Although it is completely dependent on the sporophyte for nutrition, it is multicellular and haploid individual. While the female gametophyte is developing, a similar but less complex process takes place in the anthers. Most anthers have patches of tissue that eventually become chambers lined with nutritive cells. The tissue in each patch is composed of many diploid microspore mother cells that undergo meiotic division. And each producing 4 microspores. The 4 microspores at first remain together as a quartet or tetrad, and the nucleus of each microspore divides once. In most species, the microspores of each quartet then separate. As the anther continues to mature, the wall between the adjacent pairs breaks down, leaving 2 larger sacs. At this point, the binucleate microspores have become pollen grains. Pollination is simply the mechanical transfer of pollen from its source, the anther, to a receptive area, the stigma of a flowering plant. Most pollination takes place between flowers of different plants and is brought about by wind, water, gravity, insects, or other animals. In some angiosperms, plants may pollinate themselves in a process called self-pollination. If the stigma is receptive, the pollen grain's cytoplasm absorbs substances from the stigma and bulges through a pore in the pollen wall. The bulge develops into a pollen tube that responds to chemical and mechanical stimuli that guide it through the style into the micropyle, which is the opening of the ovule to the embryo sac. The pollen tube usually takes several hours to 2 days to reach the micropyle, but in a few instances, the journey may take up to a year. The pollen tube growth is more rapid in angiosperms than in gymnosperms. Rapid pollen tube growth rate is an innovation that is believed to have preceded fruit development and has been essential to the origins of angiosperms. The nucleus of one pollen grain's 2 cells, the generative cell, divides the pollen grain or pollen tube, producing 2 sperm cells. Unlike sperm in mosses, ferns, and some gymnosperms, the sperm of flowering plants have no flagella. As the pollen tube enters the embryo sac, it destroys a synergid and then discharges its contents. Both sperm are functional, and an event called double fertilization follows. One sperm unites with the egg and forms a zygote, which develops into an embryo sporophyte plant. The other sperm and 2 polar nuclei unite, forming a triploid primary endosperm nucleus. Double fertilization is unique to angiosperms. Consequently, only the flowering plants produce endosperm. The evolutionary advantage of double fertilization remains unclear. Some suggest that by delaying the production of nutritive tissue until a point of fertilization, the plant saves precious resources. The primary endosperm nucleus begins dividing rapidly and repeatedly, becoming triploid endosperm tissue that may soon consist of thousands of cells. Endosperm tissue can become an extensive part of the seed in grasses such as corn, and provides nutrients for the embryo in most flowering plants. The endosperm nourishes the embryo in angiosperms, whereas the female gametophyte serves that function in gymnosperms. Following double fertilization in angiosperms, the single fertilization process in gymnosperm that produces an embryo, a profoundly important event occurs. The embryo stops developing. In this section, we will focus on seed development in angiosperms. In many plants, development of the embryo is arrested soon after the meristems and cotyledons, or seed leaves, form. The integuments, the outer cells of the ovule, develop into a relatively impermeable seed coat, which encloses the seed, which contains a dormant embryo and stored food. The seed is a vehicle for dispersing the embryo to distant sites. Being encased in the protective layers of a seed allows a plant embryo to survive in environments that might kill a mature plant. Seeds are an important adaptation in at least 4 ways. Seeds maintain dormancy under unfavorable conditions and postpone development until better conditions arise. Seeds afford maximum protection to the young plant at its most vulnerable stage of development. Seeds contain stored food that allows a young plant to grow and develop before photosynthetic activity begins, and perhaps most important, seeds are adapted for dispersal, facilitating the migration of plant genotypes into new habitats. A mature seed contains only about 5 to 20% water. Under these conditions, the seed and the embryonic plant within it are very stable. The embryo's arrested growth is primarily due to progressive and severe desiccation and the associated reduction in metabolic activity. Germination cannot take place until water and oxygen reach the embryo. Seeds of some plants have been known to remain viable for hundreds, and in rare instances, thousands of years. Specific adaptations often help ensure that seeds will germinate only under appropriate conditions. Sometimes seeds lie within tough cones that do not open until they are exposed to the heat of a fire, as you see here. This strategy causes the seed to germinate in an open, fire-cleared habitat where nutrients are relatively abundant, having been released from plants burned in the fire. Seeds of other plants germinate only when inhibitory chemicals leach from their seed coats, thus guaranteeing their germination when sufficient water is available. Still, other seeds germinate only after they pass through the intestines of birds or mammals, or are regurgitated by them, which both weakens the seed coats and ensures dispersal. Sometimes, seeds of plants thought to be extinct in a particular area may germinate under unique or improved environmental circumstances, and the plants may then reestablish themselves. Survival of angiosperm embryos depends on fruit development as well as seed development. Fruits are most simply defined as mature ovaries or mature carpels. During seed formation, the ovary begins to develop into fruit. It is possible for fruits to develop without seeds. Commercial bananas, for example, are fruits that lack viable seeds. Bananas must be propagated asexually. Fruits form in many ways that exhibit a wide array of adaptations for dispersal. 3 layers of ovary wall, called pericarp, can have distinct fates, which account for the diversity of fruit types, from fleshy to soft to dry and hard. The differences among some of the fruit types are shown in just a few. These are some amazing pictures too. I like this. Fruits contain 3 genotypes in one structure. The fruit and the seed coat are from the prior sporophyte generation. The embryo represents the next sporophyte generation. And finally, the endosperm is a transient triploid product of fertilization. The diversity of fruit types contributes to an array of dispersal methods. Fruits with fleshy coverings are normally dispersed by birds or other vertebrates. Like red flowers, red fruits signal an abundant food supply. By feeding on these fruits, birds and other animals may carry seeds from place to place and thus transfer plants from one suitable habitat to another. Such seeds require a hard seed coat to resist stomach acids and digestive enzymes. Fruits with hooked spines, such as those of burrs, are typical of several genera of plants that occur in northern deciduous forests. Such fruits are often disseminated by mammals, including humans, when they hitch a ride on fur or clothing. Squirrels and similar mammals disperse and bury fruits, such as acorns and other nuts. Some of these sprout when conditions become favorable, such as after the spring thaw. Other fruits, including those of maples, elms, and ashes, have wings that aid in their distribution by the wind. Orchids have minute dust-like seeds, which are likewise blown away by the wind. The dandelion provides another familiar example of a fruit. This type that is wind dispersed, and dispersal of seeds from plants such as milkweeds, willows, and cottonwoods is similar. Water dispersal adaptations include air-filled chambers surrounded by an impermeable membrane to prevent waterlogging. Coconuts and other plants that characteristically occur on or near beaches are regularly spread throughout a region by floating in water. This sort of dispersal is especially important in the colonization of distant island groups, such as the Hawaiian Islands. It has been calculated that seeds of about 175 angiosperms, nearly one-third from North America, must have reached Hawaii to evolve into the roughly 970 species found there today. Some of these seeds blew through the air, others were transported on feathers or in the guts of birds, and still others floated across the Pacific. Although the distances are rarely as great as the distance between Hawaii and the continental United States, dispersal is just as important for mainland plant species that have discontinuous habitats, such as mountaintops, marshes, or rivers. This has been your instructor, Scholar Huff.